All right, we're gonna start with a little demo-ish thing. Um, does anyone know what this is? What? My lungs?
Oh, nice. Your lungs.
Oh, yeah, no, that's good guess. Yeah. It's a CT scan of the lung from the top. Yeah. So if you look at a cross section like this from the top, it looks like this. And you would get that if you have a disease or something. Like um if you have a lung cancer, you would take CT scan and they would study it. A radiologist would study it in order to see if there are any nodules in it um, and what kind of treatment you would need. So what is wrong with this image? Has lung cancer? That's what CT scans look like. It's not, it's a, so CT scans are based on what wavelength that we talked about? X-rays. All right. So, but they're not, so when you take an X-ray, it's just one image. They shine the light, it goes through your body, and then whatever goes through gets recorded, whatever doesn't, it gets reflected, and that's one thing. What CT scan does is that they take a ton of it, right? They just go over it, and then they shine X-ray, so they get a really nice 3D, so-called volumetric scan, and that helps you, so uh, a radiologist would sit there and then go through these in depth, right? So it looks at one of these images and then it scrolls them out. It goes a little further, further, further. So it goes from the top of the lung all the way to the bottom of the lung. So imagine there are, 1,000 images stacked on top of each other that create this 3D perception. You found it?
Yeah.
What did you find?
Gorilla.
The gorilla. So who else sees the gorilla? Yeah. Everyone sees the gorilla? Here's the gorilla.
Oh, God. You are very good.
Everyone sees the gorilla? No?
A tiny outline.
The lighting is off. It's not like, all right, I'll show you what it is. So these are the legs, the face, and the arms. You see that? Everyone sees it now? So it's standing with this pattern of. So this is from a study that is called The Invisible Gorilla Strikes Again, Sustained Inattentional Blindness in Expert Observers. So what they did was that, as I said, imagine the way a radiologist would go through this is that you have a stack of images from the top to the bottom of the lung. You scroll through it, and then you spend a little time on every image, and then you go, and then maybe you attend to part of it, and then you go scroll back and forth to see what volumetric it looks like, and then you go through the whole thing. Now, what these people did is that they embedded these gorillas in the volumetric. So slice 1, 2, 3, 4, 5. So across multiple slices with different level of opacity, you could just embed the gorilla. And then they had these radiologists look through them, looking for nodules, right? They weren't looking for gorillas. They didn't know there were any gorillas embedded. Look for the nodules. And what you see here is that the inattentional blindness rate was around 80% even in radiology. So it was much higher in naive observers, like somebody like me and you would sit there and then look for nodules. But for radiologists, it was very high, meaning that as they're searching for whatever nodules they're searching, they could miss a gorilla that big in, in the image. The radiologists were much better than actually, picking the nodules, at least from naive observers. And then, but when they were actually told to look for the gorilla, they were much better. They were like, they could actually find it. Like, this is just a control experiment to tell you that the gorilla was visible if they were looking for it, but not when they were. This is an important study in attentional blindness. We talked about last time about expert attention behavior. And this is an example of that, where expert attention behavior could be really faulty. Like, you could miss really important stuff, really big salient stuff if you're not paying attention to it. All right. On that note, we're going to talk about eye movement behavior. No. So the gorilla was just one. Like, imagine you have many of these stacks, and then the gorilla was just in some corner for a few slices. So it's a hard task. That's why it's so high. Like, if it was just this slice in front of them, maybe they could have detected it easier, right? But if it's a volumetric thing, it's like there's much more opportunity. Because, for example, you're scrolling through this and you see something that you want to pay attention to here, and then you go back and forth, and then you don't, maybe it's nothing, and then you scroll forward without even looking at this part. All right. So eye movement behavior, we talked a little about this last time. I think when we were talking about covert and overt attention, and I talked about how in the context of the study that we looked at in bias competition theory, I was covert attention. The eye was fixated in a certain area, and then there were objects that the monkey was supposed to attend to without moving their eye. That's what we call covert attention. And this is our ability to attend without moving our eyes. We could covertly attend to a lot of things. We could covertly attend to, auditory stuff. We could covertly attend to a lot of things. But when it comes to vision, it's about not moving your eye. Attending to an object in the periphery without changing fixation. Overt attention, on the other hand, is actually you are, moving. You're moving your body in order to get a better sense of what is the sensory input, right? If you're trying to hear someone better, like, you could get closer to them, or you could turn toward them. If you want to see something better, you would look to them, right? A lot of our actions in life, in a daily life, is about just bringing something to our attention, right? When you move your body, when you move your head, when you move your eye, a lot of that has to do with trying to bring something to fovea or to your center of your attention. That's the difference between covert and overt attention. So we talked about why we move our eyes, because we want to bring something to our fovea. We want to make sure that the light coming from a certain object reflects on the fovea. In this case, if you have a fixation point and you're looking there, that's where the reflection from that point falls on the fovea, and then the reflections from all the other points fall on the periphery. The fovea is special in a lot of ways. We went through this. This is just refreshing your memory. The fact that there is a high distribution of cones in the fovea versus the high distribution of rods in the periphery, it gives you a much better resolution because the convergence is smaller in the fovea. The ganglion cells part ways, so the light doesn't scatter when it hits the fovea, and the fovea is right on the optical axis of the lens. So all of those things make sure that the light that reaches the fovea, it doesn't get degraded or scattered or gets averaged out too much. So all of those things allow the fovea to be much higher resolution. Another way of saying it is that to have a very high visual acuity. Again, these are all the stuff we talked about this. Now, what is the effect of that? What is the effect of having much bigger averaging windows in the periphery? Is that when you, for example, here looking at this letter in the middle, letter A, you could actually read the peripheral letters, right? You could actually read this S in the periphery. But the reason you're able to do this is because those are magnified so much. You have to make things so much bigger in the periphery in order to be able to read them. Versus when you get closer to the fovea, you could actually read things in the periphery when they're not as big. Does that make sense? You get higher and higher resolution. Higher and higher resolution means that you could resolve more details. And if you can resolve more details, you could read the smaller things. Make sense, everyone? The acuity decreases with visual eccentricity. So the acuity decreases with increasing visual eccentricity, it should say. So eccentricity means how far you are from the center. So if there's a higher eccentricity, it means that you're far from the foveal point. And as you get farther, you have to get bigger and bigger to be able for it to be legible. So eye movement behavior is mediated by a different pathway than the one that we talked about before. We talked about the geniculostriate pathway. This is the pathway that takes the visual information from the eye all the way to the primary visual cortex through LGN. This is a pathway we talked about. But the other pathway is the tectopulvinar pathway. This one, some of the ganglion cells actually branch off from the other pathway and then go directly to an area called superior colliculus. So superior colliculus is an important part, plays an important role in eye movement, planning and execution. And it's a very, very old brain area, in a sense, old in a sense of evolutionarily old. So before animals had neocortex, right, lizards and frogs and those animals, this is their entire visual system. The input from the eye goes directly to superior colliculus, and based on that input, they make actions. So they don't have the neocortex that mammals have. And that was their entire visual system. But in humans, we build the entire cortex, like we have the entire cortex on top of that. So eventually the input from the superior colliculus also projects back into the cortex. But superior colliculus still plays an important role in eye movements. It's a very deep area too. So as you could imagine, evolutionarily, so these areas were built on top of each other. So the cortex, which is the oldest, is on top of everything. But deeper down, you could find like, you know, older areas like superior colliculus in the brain stem pretty much. So an important property of the superior colliculus is that it has a retinotopic map. That you shouldn't be surprised because it's a visual processing area. It's likely to have a retinotopic map because it gets its input from the retina. And because retina is organized in a certain way, superior colliculus is going to just continue that organization. People have found out that superior colliculus is involved in eye movement planning, in the action planning, because when they stimulated different parts of this area, this surface, they saw eye movements of different degrees in different directions. So let's first talk about what degrees are. I think I explained this at some point, right? Degrees are, when you talk about eye movements and actions and visual fields, you talk in degrees because that's how we see the world, right? So the whole world would be 360, maybe, you know, minus 90 and 90 here. But according to this graph, maybe this is around 60, 70. So it's saying that you could actually make eye movements as much as 70 degrees to the right and 70 degrees to the left. That's the idea that we're saying. Now, if you stimulate certain area here, then you're going to get, for example, an eye movement. So if you stimulate this part of the superior colliculus, the monkey will make an eye movement 10 degrees vertically toward top. Then you could say, if I stimulate this part of the superior colliculus, the monkey makes a eye movement 10 degrees to the right. So knowing that this is a retinotopic organization, if I stimulate this area, what would I get? Like up here, what would I get? Diagonally, right? I would get something in between. If I stimulate this area, what would I get? Yeah, so you get a little bit to the right. So movements that are similar in a space gonna be mediated by tissues that are close to each other in the brain. Does that make sense? So alternatively, when we were talking about perception, we were saying cells that perceive similar areas in a space are close to each other. In this case, it means that actions that are similar in the space should be programmed by like close by tissues in the brain, something like that. Now you could see that this much of the tissue is for stuff that are 10 degrees, and then when you go all the way out, 50 degrees or this much of the tissue. What does this remind you of? Yeah, so foveal magnification, right? Meaning that, in this case, what would that mean?
So here it seems that like the areas that are, I guess, less than 10 degrees, they're overrepresented.
Yeah, so a lot more tissue is allocated to programming shorter eye movements. That's what that means. That the reason being you want to make more accurate eye movements when they're smaller. So when more tissues is allocated to something in the brain, almost always it means that you're more accurate at doing it. Does that make sense? So when we talked about attention, and attention, for example, magnifying the activity of an object or allocating more tissues to an object, it could lead to a more accurate representation of that object. And that's the currency in the brain, right? You want to be represented more. So there is another form of foveal magnification here in action space, which means that there's just more tissues for shorter eye movements, or what we call saccades. And these shorter saccades are just more common. And that's how, brain is organized. It's a trade-off. If you have more common actions, you want to be able to do them better than actions that are not as common. That's just how the trade-off would work. So yeah, as I said, if you stimulate somewhere up here, then you're going to get an eye movement that is diagonal. And that's how it works. I hear a little bit of my voice coming back, but that's fine. But also eye movements are represented using a distributed code. What does that mean? That means that so when you stimulate a specific area here, you're not stimulating just a single cell, right? Cells are connected to each other. Any form of a stimulation really has a diffused effect on the activation. And for example, what really causes an eye movement in this direction is not just a single cell, but it's a population of cells. That there's a population of cells collectively coding for that movement. A lot of them are coding, we're going to talk about this a little more. A lot of them are coding for horizontal rightward movement. A lot of them are coding for vertical upward movement. And together, they're coding for a diagonal movement. But it's a population. And this should remind you again of what we talked about before, which is the distributed coding, right? So we saw examples of distributed coding in a few different lectures. And the idea is the same thing, is that if I want to code a movement, I can't just rely on a single cell, right? There has to be a population that is coding for that. There should be a population that is coding for that. And in this case, this is also using distributed code.
They were kind of They altered by every like 10-degree angle and they were kind of together, organized together. Is this organized the same way or is it random? Like the orientation?
So the orientation here, again, this is a schematic, so
Uh-huh.
don't take this too serious. In the superior colliculus, it's harder to quantify this. In the motor cortex, I'll talk about that more. This
Okay.
orientation, selectivity, and how they're organized. It's easier to discuss that in the context of cortex. This is a subcortical area. These schematics are a little simplifying, but as you said, it's like there's more gradedness to how their orientation changes. There is also a laminar, so-called layered structure to superior colliculus. Some of these motor cells, cells that when you activate them cause eye movement, are deeper, and the higher areas are more visual. But that's a different organization compared to the cortex. It's still layered. To do a little demo again. So you're gonna see a bee, and you're supposed to just follow the bee. Just look at the bee and follow it. All right. Everyone sees the bee. All right, let's do it. So now you saw the bee, and then you followed the bee, right? Now the question is, did you notice anything changing in the periphery? No, but let's rewatch it and just watch that flower that the bee started on. So what happens to this flower? It's getting blurry, right? So, um, so what is this showing? This is showing that as you were watching the bee, you were getting farther and farther from this flower. And the experimenter knew this, right? Because they asked you to follow the bee. So what they were doing is that they were just making the flower blurrier and blurrier depending on how distant you were from it, and you wouldn't notice it, right? And that's the experiment, is that you don't know that you're not getting all the information in the periphery. It's just that when you do an experiment, you could show that when you make it blurrier, people wouldn't notice it because their representation of that area is already blurry, so they can't really understand that, oh, something is getting blurry out there. Does that make sense? So it's just a display of um, how degraded the information in the periphery is. Uh, if you're wearing glasses, you probably, if you don't have your glasses on, you see the world in a blurry way. Is that true? Like you don't see like fine-grained details. And imagine everyone else has that in their periphery, where everything is degraded. Again, you don't know, you're not aware of it, but when it's an experiment, you could show it. It's the case. So eye movement behavior is important in a sense that you could really tell what people are paying attention to. Now, some of you read the paper on eye movement behavior and autism, and the and you saw that you could do experiments based on what people are attending to, based on where they're looking. You could learn something about, you could come up with diagnostics and stuff like that, and understand their what kind of information they're gravitating towards, right? So attention is important for those tools. So one other thing that people have used it, and you may have seen it, is more of a human computer interface using eye movements. And we're going to talk about brain computer interface in the second part, but in this part, I want to show you a demo of a human computer interface. And this feels a little like an ad for this Toby thing, but try to ignore that part.
What do you think, Francie? What's going on? I'm dreaming of drawing.
He's not one to complain, you know, he's just kind of taking this all in stride. So I don't think I really noticed the difference until he got the eye gaze. And at that point, it's like, whoa, okay, you know, yeah, he's back, you know, that's he's got this device now and he has a way to get it out.
His eyes got tired, but he
Why is there someone listening to the talk somewhere? Or is it coming through mine? Oh, you're not. Is it my speaker that is reflecting?
It might be, so maybe turn this volume down.
But then we wouldn't hear it, no? Or is it coming through there?
And then if you go into the sharing. Oh, no, I guess you would have to share sound. No, you're right.
That's fine. Don't worry about it. It's not too bad. All right, thanks.
he was smiling and just another man.
And then
So-
suddenly he was just, you know, producing image after image. He was posting on Facebook. He was interacting with people.
So this is brain-computer interfaces based on eye movement have been have been in development for a long time. So they're much more sophisticated than the other example, the brain-computer interface. These are human-computer interfaces. But brain-computer interfaces that we're going to look at are not there yet. But in this case,
So yeah, it really did make a huge difference. And
if you look
especially-
at it, so the person looks at something, right? Looks at a key on the keyboard, and then if they hold it long enough, it's gonna register it, right? Because you don't want to register everything they're looking at, because sometimes they have to look for something. If they're looking for a specific key, they can't register everything. So they look at something, and then if it's enough time, it registers it. Now, you can imagine.
So away
Uh,
from a lot of the people that he would have contact.
You can imagine that with something like this, if you're really good at it, if you practice with it, then maybe you could type, 10, 12 words a minute or something like, you could really write a book, and people do write books in a year or so, when they have a device like this.
Contact with, you know, just staying in touch with people, kind of discovered that he was that statement up on his store. And
But yeah, there are now technologies that are sophisticated based on these sorts of eye movement based interaction with the environment. Like a lot of people, when they get sclerosis or other muscle related injuries, they still like, I feel like the eye muscles and the eyelid are like the last thing to go. So they are still able to communicate through that. All right. I wanted to show you that. So we talked about attention and eye movement. So this figure is here just for me to tell you attention is studied also as a network, right? So when you have a complex system with a lot of moving parts, attention is really not just an effect that you see, you observe in one area where it's like the magnification of information for something or the over-representation of something, but rather the way it is gated along the hierarchy in the entirety of the network, right? So if I'm attending to something, there is a attention network, frontal, parietal, or other network that people have mapped out, where you see the information about this object being communicated across the brain. So people think of these as more of a network that we talked about. For example, in parietal area, you have that if you have damage, you have these sorts of neglect effect, which is attention related effect. You have areas in the front that also modulate attention in the frontal area. You have, temporal, frontal on this side, parietal on this side. Sorry for it's a weird looking brain, but the features are down here, which is the temporal area. I said superior colliculus is really deep, so you see it as this color, which means that it's underneath everything. So it's a combination of a lot of areas that together create the attention network. And I think we're not going to get the chance to talk about these sorts of network level properties in the brain, like there are, for example, resting state network, which is, when you put people in a scanner and they're not doing any tasks, they're just daydreaming. When they're daydreaming, you see activation of certain areas that just kind of go back and forth. It's like a whole network that looks complicated and it's just active. And they use that, you could use that to predict certain things about people, like how creative they are, or some pathology, or something else. You could actually use the activation in the resting state network to predict those things. But yeah, we're not going to get a chance to talk deeply about networks, like what networks are in the brain and how they communicate. We talked about pathways, like visual pathways, and those are also networks. But I think there's something interesting in understanding a network of area across time that you could read into if you're interested. Okay, so more broadly, talking like attention behavior, eye movement is very common, as I said, like we make eye movements a few times a second. And it's an important behavior, but it's not, it's a somewhat simple behavior. We believe that it could be controlled if you have a map of this space, like a retinotopic map, and you have some activation somewhere in that space, you could actually, program a circuit to that. And you could imagine those activations could come from something like a saliency map, like the one that we discussed last week. Like you could have a saliency map that areas that are really high contrast, or for us, maybe areas that have faces and those sorts of things. So then there is motor areas where it's actually for controlling all the limbs and all the muscles that are in our body. We talked about the topography of areas that are represented in the cortex as action. And we talked about how people could find these. Do you remember the tool that was used to map this out? Yeah, so what was it called? No. It was called TMS. TMS, where you could actually, as you said, you could shock different parts of the cortex on the surface, and you could see movement of different body parts. It's not a clean mapping, but you get this sort of broad mapping of areas to different body parts. And you have like the hand, you have the hips, and the face is overrepresented, as you can imagine, and hands are really, really overrepresented. Because, well, we have really dexterous hands, right? And that's part of our evolution and civilization, like having the opposing thumb and being able to hold tools and use tools. That's a big part of our evolution.
Like really quickly, like in the sort of like peripheral like motor system, like is that because like here it's like you're shocking like the central nervous system, right? Essentially, and it's like sending something to like the periphery. But like if the periphery is like affected, like you get like your knees tapped at the doctor's office, is that going to like the same part of the brain?
Well, if you have
Or if it's in the periphery, does it just sort of stay to this and not appear to the center?
well, it depends, right? So a lot of nerve cells end up going back to the brain, right? So if you're being touched like on your knees, then obviously that would be sense, and it goes back to the brain, and you have the somatosensory areas that process that information. But also we have proprioception, right? Everything that we do, we have a way of knowing where our body is in a space. And that's different from what you're describing. That's like volition, right? You know, when you move your body, you know that you moved it a certain bit. But yeah, I think a lot of these signals would have to go back to brain, because we talk about this later. It's all part of a control loop, right? You have to, when you act in the world, you have to know what you've done to correct it, So it's always that there are a lot of feedbacks that you get, right? A lot of time the feedback is visual, that I could see where my hand is, so I could move it up a little bit more to grab the mouse. But a lot of time, even if I close my hand, I kind of know how much I have to move, and that's through proprioception, right? You know, you get information about where your limbs are and where your joints are and all those sorts of things. Okay, one way of mapping this is to talk about, let's talk about some motor areas, right? So in this graph, this is what's called the motor cortex. It's like any cortical area, it's on the surface, and you have premotor areas too. And when you record from this area, people have found a pattern like this. So imagine the monkey is sitting there. There is a lever in the middle, and the lever could be moved either to one of the points in the periphery outside, or it could be brought back. So that's all the actions the monkey can do. The monkey can take the lever, move it up, move it back. They can move it to the right, move it back. Now, this is what I'm showing on the right is recording from a single cell. What do you see in this single cell? So first of all, what are we looking at here? What is this? What's that? The firing, right? So what is each row? Usually when we show it this way, what is each row? Different trials. And each bar is an action potential. So you always repeat the experiments for many trials because neural data is noisy. So in order to get a clean data, you have to repeat it many times. And then you look at the average activity. But when you look at this, in which directions does the cell activate? No. So these arrows are showing the direction of movement. So this is showing all the movement that started from the center and went to the different positions, to the 8 positions that are in the outside. So when you look at this, you get a sense of, well, there is a certain direction that this cell gets really active when the monkey moves, right? And that's the downward direction. So when the monkey is moving the lever from the center down, it gets really active. Not only that, when you look at the bottom, it's the same cell. When the monkey is moving the lever from this peripheral location to the center, it also gets really active. So when people looked at this, they were like, okay, maybe what a motor neuron cell is doing is that it's coding for the direction of movement. Does that make sense? Yeah.
What's the difference between the red and the green?
The red and the green is just a visualization of what we see as being high activation. It's not like, yeah, there's not a difference between them. It's just the visualization to highlight that these are the directions that have very high firing rate versus the other direction. So in this case, when you, all these movements are the same length, but they're in different directions. And what we see is that even when you're coming from peripheral location here or from there to here, in both of these cases, you have really high activation. So it's kind of the direction of movement downward that this cell really likes. So when we were talking about visual cells and we were talking about the direction selectivity, as you asked, in this case, also people have found that there's some direction selectivity, but it's a different type, right? Back then, we were talking about the direction of visual information, like direction of the orientation of the bar. But in this case, or direction of movement of the bar, in this case, we're talking about the direction of action. The neuron is most active, greatest density of dots, meaning action potential for movement in the downward direction, red arrows, regardless of its starting and final locations. So regardless of those two things, we see higher activation in the downward movement. So we could then look at the selectivity of a single neuron, right? So when we talked about the selectivity of neurons in the case of visual perception, we say, well, a certain V1 simple cell is coding for the orientation of the bar. But if you just move it a little bit, you still get a little activation, right? When you move it a little bit more, you still get a little activation, a little less. So it was graded, right? It wasn't like that the cell only codes for one direction, and then as soon as you turn it a little bit, it becomes completely unresponsive, right? So there is this gradedness to the activation. And in this case, that's the case too. So let's say there is a cell that is highly active when you go in this direction, 180. Now you could show it as 180, and the response rate, the firing rate of the cell is at maximum when the lever is moving in that direction, when the hand is moving in that direction. But when it's moving to, let's say in this case, nearby, and imagine 0 is the opposite of that. So on 0, it's very low response. But everything that is close to 180, in this case, you know, I guess 145, or it would be 135. 135, then you would have somewhere in between, some firing rate in between. Okay, so the idea is that it's not that the cell only is active for one direction of movement and it completely goes quiet for the other ones, but rather there's a gradedness to the response. So you could actually, I'm not sure if we talk about these sorts of tuning curves in the case of visual response, but one way to show that, you could have just had an x-axis that shows all the different orientation, and then a y-axis would show the firing rate, and for some cells you would have, for all cells, you would have this kind of a graded tuning. Okay. How about we take a break for 10 minutes and then start at 7:08? Okay, so looking at this, you see that cells could be active to different degrees depending on the movement and what they prefer, right? So in this case, there was a question earlier, you could see this gradedness where the cell is active maximally for a certain direction of movement, but that falls off gracefully when you're making different movements. So the cells is not going to go quiet completely, but rather slowly fall off. Now, what if we want to actually plan a movement, right? What would, knowing that there are different cells that are maximally active in different direction of movement, how would that population of cells code for a movement in a certain direction? Well, if each of them, let's say these are two different cells, one of them is coding for the upward movement, another one is coding for the leftward movement. If they're both active, then maybe you're coding for, you know, up leftward movement, right? But it also depends on how active each of them are. If this one is really active, the upward movement is going to have more firing rate behind it. And the leftward movement maybe doesn't have that much firing rate behind it. So the direction of movement changes. So this is a form of, again, population coding for movement, where it depends on which cells are active and how active they are. So if I want to make an upward movement, the way this movement would be programmed in the motor cortex, it's by having a lot of cells that have that movement as their preferred movement be active. Does that make sense? So, for example, imagine there are, you know, I'm standing here and there are, you know, people around me and I'm tied, and let's say I'm tied to a rope and each of them have, you know, one end of the rope and they're pulling me in different directions. Does that make sense? So those are the cells and they have preferred directions. Now, if I want to go in this direction, it would mean that the people who are on this side are pulling me stronger than people who are pulling me on that end. And these two directions are kind of canceling each other out. So this is a form of population coding for movement. It really depends on the population, how active they are. So in this case, so what this figure is showing that when you're trying to make a movement in this direction, this could be how the 200 neurons are acting. Now, each of these lines is the preferred direction of a cell times how strong it's firing. Does that make sense? So you have 200 cells, for example. I don't know what the exact number, but imagine there are 200 cells. This one has this preferred direction and it's really active. This one has this preferred direction and is not as active. You could see why this pattern of activation across 200 neurons would pull the hand in this direction. Does that make sense? So this pattern of activation across 200 neurons would pull the hand in this direction. This pattern of activation across the 200 neurons would pull the hand in this direction. So if the 200 neurons are active, in order for you to move in the right direction, what does it mean? Well, the cells that are really like that direction of movement should be really active because they are connected to the muscles in a way that when they're active, the hand moves in that direction. So that it comes again from their connectivity. The cells here are connected in a way. So in this case, if you want to have a leftward movement, you have to have cells that really like leftward movement to be active because, well, when they're active, they command the muscles to move the hand in that direction. That's the idea. In this case, again, this pattern of 200 neurons, if they're active in this way, the hand moves in this direction. So this is, again, a form of population coding. Each of these cells have a vote. And depending on, not a good metaphor these days, but each of these cells is pulling you in a certain direction. And depending on their ratio of their activation, you could move in a certain direction. The reason is that, well, each of them is connected in a different way. And when they're active, they push your hand in a certain direction. And when the population is active in a certain way, you move in a certain direction. Does that make sense, everyone? Yeah.
Do you know what causes conditions like dyspraxia or dysgraphia?
What is dysgraphia?
Dysgraphia is a retardation of the fine motor skills.
I see.
So like you can't read your own handwriting.
You cannot read your own handwriting.
Yeah.
I'm not sure. Is it a muscle issue or is this a more of a command issue? I'm not sure.
I don't know.
Yeah, I'm not familiar with that.
that.
So I think I'm going to show a few examples, but those are muscle issues where your muscles are getting weaker, or let's say you have an accident and there's a damage to your spinal cord, but the brain is still intact, right? So if the brain is intact, then you should be able to, create these decoding devices that I'm going to talk about in a little bit. But I'm not familiar with those cases. It could be, again, the issue could be that the brain is, going through something that cannot command properly, or that the muscles having an issue that cannot execute commands. So this is a form of population coding for direction of movement. So it's stuff that like to move you in a certain direction would be more active if you want to move in that direction. That's the gist of it. It's not complicated. In this case, there were 251 motor neurons, and I didn't set around 200, but these are just gets you more description of what that is. Now, knowing that, well, cells are coding for the direction of movement, you would think that we would be able to decode, and I'll unpack what that means, decode what the intention of movement is. Right? So if you try to move in a certain direction, I could look at the activity. Let's say I put an electrode array and try to predict the activity. I record the activity and I try to predict the direction of movement, and I should be able to do that based on this finding. So that is, and some of you read the paper on the brain-computer interface for the assignment too. That is the basis of a lot of brain-computer interface devices, right? So in many cases, you can imagine an individual that is tetraplegic, where they have the spinal cord connection to the rest of the body is severe. So because of accident or stroke, and they cannot really move their limbs anymore. And over time, they lose their muscles. But what you're doing is that, what if you're saying, I take this electrode, and these are called Utah arrays, and they're usually around 64, but there are bigger ones now. And you could place this, and it's really small. It looks like a needle bed or something, but it's really small. It's, compared to a penny, it's really small. And you could see it in an MRI where it's placed. And where would this is a motor area that we talked about. And the motor area is a cortical area. So it's on the surface of the cortex. So what you do is that you could open up the skull and then place the array into the cortex. You could just, plant it in there. And you could see it on MRI. Where would that be? That's the left motor cortex of this individual, and you could it's placed right there. Why is it the left motor cortex usually? Exactly. Because when you want to read these commands, you want to read it from an area that is really dexterous. Like the neural firing is really, really complicated and has a lot of information. And for a lot of people, because they're right-handed, the area that has most information is the hand area of the left motor cortex. Does that make sense? So in the left hemisphere, if you go to the hand area, then that's where you really get a good signal. And that's the signal people use for decoding. Now, so when we talked about, I'm going to step back and talk about something about information and why this is doable even. So you could say that when you have a complex system, a lot of time, you don't have to understand all the complexity of the system in order to predict how two things are related to one another. For example, I could say the color of the leaves carries information about the month of the year, right? What does that mean? That means that, let's say you were, I don't know, in a coma, and then you woke up after a few years, and I ask you, what months do you think it is? Well, you wouldn't be able to tell me what month it is, right? But then I say, look outside and look at the trees and tell me what month it is. So initially, you have no way of knowing. You have no information. It could be uncertain. It could be any of the 12 months. But when you look at the trees, then you could narrow it down maybe to 2 months. Be like, hey, they seem yellowish, so it must be maybe October or early November. So you're predicting one variable from another. You're predicting what month it is from the color of the leaves. That makes sense? So there are two variables. Now, how are these two variables related to one another actually? If you wanted to map out how these two variables are related, can you imagine how so why would the color of the leaf in November be yellow? Well, that story is really complicated. You have to know the planetary movement and the Earth and the orientation of the Earth versus the sun, and this part of the month there's not enough sun maybe, and then there's photosynthesis and this type of tree moves. You see what I mean? If you actually wanted to know how a month of the year is related to the color of a leaf, that would be an extremely complicated story if you actually wanted to understand it. But you don't have to a lot of time. And that's the beauty of it, right? You don't have to know all those things. All you have to know is that you have seen different colors of the leaves in different months. Does that make sense? So you could relate two variables to one another. You could predict one from the other. Technically, there's a mutual information between two things. And you could detect that, you could learn a mapping from one to another without worrying about the entire complexity that relates the two. And a lot of time in life, when we make these heuristics and know one from the other, we're not really worrying about the complexities of the world. That's a long story, but the point is, this is the case when we do decoding. We're saying that the reason you code a lot of information in the brain, in this brain area for movement and the entire machinery of the brain and how it works and all of those things, I say forget about all that. All we need to know is that I ask my man here to intend to move the cursor up, just intend. And I record from their motor area. I tell them, and they do that many, many times. I tell them to intend to move their hand down. They cannot, they're paraplegic, but I ask them to do it. I ask them to intend to move to the left. I ask them to intend to move to the right. And I record all this information. I record what the neural activity is. Now I have two variables. I have the intention, which is which direction they're moving, and I have the neural activity. And I can just relate the two variables. Now, next time this person is sitting in front of the computer and they just intend to move the cursor up, well, now I could record the activity and then go check which activity that I recorded is most similar to the current activity. And then based on that, I know, well, their intention is to move up. Does that make sense? And that's what decoding is. Now, does that work all the time? Not really. For example, let's go back to my example of a person relating the months to the color of the leaves. What if a person grew up in Toronto and learned some mapping between the two things, and then we move him to Jamaica? Does his mapping function work in Jamaica or some Caribbean places? No, why not? Yeah, right. So that's the problem. If you do not understand the complexities of the world, whatever model you have of relationship between variables is not going to hold over time, because context changes, the world changes. And that's also the case with a lot of these brain-computer interface devices, right? A lot of times, so brain is an organ, and there are a ton of veins in it, right? Veins in it, because as you know, we talked about fMRI, for example. fMRI records blood oxygenation in the brain. So every time that the heart pumps, brain moves, right? There's a little pumping of the blood into the brain, and then it just, every time. So the brain moves, these electrodes move with every movement of the brain. So they record from different cells every time, and then the neural activity kind of changes, drifts over time, and cells die, new cells are born all the time. So the world changes so much, and the brain changes so much over time, that you have to recalibrate this thing often. And that's the issue with a lot of brain-computer interfaces, that they're not reliably available unless you continually recalibrate. What is calibration? The process of what I described, that you have them perform different actions, intend to perform different actions, and then you record that data. That process is called calibration.
Change, or the leave example, like you're changing location. Well, and that, but what accounts for the changes generally? Like why do you have to recalibrate, like with the brain, for example? Is it like the temporal aspect where like the brain changes like over time, or is it more related to like the brain's in a different the body's in a different context? Like is it like the more like the spatial or the temporal?
All of those things, right? So I think a lot of it has to do with just the activation not being, so the brain is just, there's a lot of drifting in the brain, and that's a complicated thing that we're not going to get into. Meaning that you could have activation of a certain cells, and then maybe a few weeks later, the neighboring cells have similar activation, and this one kind of changes. Like kind of the selectivity changes a little bit over time, and that's a problem. But also a lot of other things. Just the, as I said, physical movement of the brain itself with the blood every time. Or when you're in a different environment, when you're in a car and they're speeding, the brain kind of moves. All of those things. But also just the person changes. As I said, a lot of cells die, so you're not recording from the exact cell, so the pattern kind of changes. Now, these sorts of mapping that I talked about, the mapping from, let's say you collected some pairs of things, pairs being the intention to move in a certain direction and the neural activity. It's not a simple relationship at that point, right? Even that mapping would be a neural network, for example. You learn a neural network to map from the recorded brain activity to what the intended movement was. It's a complicated mapping. Because it's not as simple to just look at this pattern of activation and be like, oh yeah, that's most similar to this one or that one. It's a complex spatial temporal pattern. So yeah, that's decoding. Now this is in contrast with encoding. What is encoding? Well, encoding is what we talked about in the context of perception. When you were looking at something, the neural activity encodes information about that object, right? A subpopulation could encode information about the color of that thing. Well, what does that mean? Well, it means that the change in the activation carries information about the color of that object. If you change the color, the activation is going to change. So naturally, you should be able to decode that too, right? And some of you read that paper on attention decoding the stuff, and that's the idea, where they were decoding the different properties of the object from that encoded representation in the IT. All right. So that's encoding, right? When the response of a cell carries information about the visual stimulus, that's encoding. And when you read out that information from the brain activity, that's decoding. Okay, so these are the components of a brain-computer interface, of a BCI. So you have the brain here, and you could record from the motor areas, or you could record from the brain stem, depending on where the damage is. So you get the signal. The signal is the neural activity that we talked about. You process this signal. The signal processing itself is a complicated process, right? You can't just look at a neural activity. No, you have to look at it. You say, okay, what are the features of this neural activity? What patterns there are? And then that pattern recognition says, okay, I think this neural activity means upward movement. This neural activity means rightward movement. And then you create a control signal. You can control a robotic arm. You could control a cursor on the screen. And then you get feedback, right? And this feedback is really important. The person has to know what is the consequence of whatever they're doing. And then they could adjust themselves, right? When you put people in a control loop, they can adjust their behavior and they adjust their mindset and then change stuff. All right, let me show you a demo of this in a monkey.
This is Pager. He's a 9-year-old macaque who had a Neuralink placed in each side of his brain about 6 weeks.
So in this case, in a macaque monkey, a lot of time when you do experiments, right, you create a chamber, right? You cut through the skull, put a chamber, and then you put the electrode through that. In this case, they actually created a wireless system so the macaque can easily move around. So they planted the chip under the skull, and they attached a wireless device to it, so you could read the signal wireless.
Weeks ago. If you look carefully, you can see that the fur on his head hasn't quite fully grown back yet. He's learned to interact with a computer for a tasty banana smoothie delivered through a straw. We can interact with the Neuralinks simply by pairing them to an iPhone. just as you might pair your phone to a Bluetooth speaker. The links record from more than 2,000 electrodes implanted in the regions of Page's motor cortex that coordinate hand and arm movements.
So this is from 2,000 electrodes, right? So this is a much bigger electrode array. Now, what are they doing here? The monkey is moving this joystick, right? And then at the same time, their activity is being recorded. So these are the pairs that I was talking about. With every movement of the hand, there is a neural activity. So they could pair and they say, okay, when the monkey was moving their hands up, this was the neural activity. When it was moving it down, this was the neural activity. They could record all that information.
Neurons in this region modulate their activity with intended hand movement. For example, some might become more active when he moves his hand up, and others when he moves it to the right. By recording from many neurons and feeding their activity into a decoder algorithm, we are able to predict Page's intended hand movements in real time. First, we calibrate the decoder by recording neural activity as Pager uses the joystick to move a cursor to targets presented on the screen. As he's playing this game, we are wirelessly streaming, in real time, the firing rates from thousands of neurons to a computer. Using these data, we calibrate the decoder by mathematically modeling the relationship between patterns of neural activity and the different joystick movements they produce.
So in this case, just to have the monkey go step by step, they just unplug the joystick. Now, in this case, the monkey doesn't know it, right? They're still making the movement as if, but what they don't know is that now the activity is just being recorded and decoded to movement. So the monkey thinks that they're doing this, through the joystick, and they continually move that. And that helps the experiment because as they move it, the activity becomes more similar to what they initially recorded. And it makes the decoding
After only
easier.
a few minutes of calibration, we can use the output from the decoder to move the cursor instead of the joystick. Page still moves the joystick out of habit, but as you can see, it's unplugged. He's controlling the cursor entirely with decoded neural activity. Our goal is to enable a person with paralysis to use a computer or phone with their brain activity alone. Because they wouldn't be able to move a joystick, they would calibrate the decoder by imagining hand movements to targets.
Yeah, right. In the case of this monkey, it's easier because they could actually make the monkey do it. But in the case of the human, it's easier in a different way. You could ask them to intend to do a
One of
movement.
the things the Neuralinks allow Pager to do is to play his favorite video game, Pong. To control his paddle on the right side of the screen, Pager simply thinks about moving his hand up or down. We've removed the joystick altogether. Now that he's up to speed, let's increase the difficulty and see how well Pager can play with the Neuralink.
So what you see here is,
As you can
again, the entire process of building a brain-computer interface is this, where you want to record from certain areas, and then you want to map those recorded neurons to intended movement, and then you calibrate it, you build your system, and then you put them in a loop. And that's all there is. In humans, I'm going to show you just one last video for humans.
Is imagine moving your right arm, for example, to the letter E.
So imagine this is a patient and she cannot move any of her limbs. And
in addition to that, we're also able to detect when you wish to select that letter T. Our participants were able to type.
If I can show you what a chamber looks like, it looks like something like this.
100 tiny electrodes that just.
So it's on this skull, you create a chamber, it's a metal chamber, because if you don't, the skull grows back. Like bones always grow back. So you have to put a metal thing so it stays open.
It's on the surface of the brain. And it's
what? Yeah, so the monkey, in the lab, when you do neurophysiology experiment, you create a chamber and you put the electrode through that, and then there is it has a screw where you could close it off. But in this case of the other monkey, they actually did the surgery, they planted it, and then they let the monkey kind of heal from it because it's a wireless system underneath. But I don't know what the mechanics of it are.
Able to pick up on the electrical
this is-
activity of individual brain cells or neurons. We record tiny signals from those neurons, those brain cells, called action potentials. We can then take those signals and decode them using a computer. We can manipulate them once they're in digital form like any other data.
So in this case, like the process of calibration would look something like this. You know, you have a circle in the middle, another circle comes up, and they're supposed to, with their thinking, they're supposed to intend to move it to that spot. And then another light comes out, they intend to move it in that. So they collect all this data, which maps the neural activity to that intended direction. And then later on, they decode it. All right, this is a human example. So these are the main components. So these are the main components of that system. So a lot of time, as I said, these models, these systems do not worry about the complexity. They're not worried about all the ways that the activation came about, what the planning is. All they're trying to do is to make sure that those two things map to each other. Now, a lot of time in life, as I said in the example of the leaf and moving and the world changing, a system like this is not reliable, right? It's not going to last you too many weeks, like even 2 days without recalibration, because that's how much things change in a living organism. So you have to continually recalibrate them. And it's also a bit deeper point here where in order to have a more robust system, it's important to understand the complexities of the system. Like if you understand the complexity of a system, then you're more likely to continue being useful. Your model of the world continues to be useful when you understand the complexities of the system. All right. You could also imagine cases where you could use this for rehabilitation, meaning that a lot of time when you cannot move your muscles, you lose that muscle tissue, and some BCI system try to just take the signal from the brain and then stimulate the muscles that you already have in order to kind of bypass that whatever stem injury, brain stem injury that people have, and you could rehabilitate like an arm or something like that. I'm not sure. It's an active research area. They're not really devices that work right now. Any questions about this? It's just good to know. So as you'll see, there are going to be a lot more decoding devices in our lives, I think, right? Meta, Facebook parent company, have recently released the wristband for controlling VR. I don't know if you've seen it. So they basically put a wristband. It's a sleek device at this point, and it records the motor signals in your arm. So if you do movement with your hand, it's going to pick that up, and then it's going to transmit them to whatever screen you're looking at on your face, right? So if you have glasses, you have a screen, the screen is doing something, and you want to maybe, I don't know, turn up the sound or turn down the sound, and you could make this movement, and it just picks up on all those movements. So these sorts of interfaces, neural interfaces, going to become more common in the way we interact with the environment, with the devices that we have. And so it's good that you know about them. So moving on, I just want to introduce something that you may see out there, and they're called mirror neurons. And this was something that was, I think, found a couple of decades ago, and it had a moment in a sense that people thought, oh, this could explain how perception is related to action and all those sorts of things, but it didn't really deliver. But it's good that you know about it. What they are is that when the monkey is maybe cracking peanuts, or when the monkey sees someone cracking a peanut, or without hearing the sound even, or just hearing the sound. I think that's this porous thing is supposed to show that you hear the sound or something, or this is maybe you just see it, I think. And so in all these cases, they represent some same thing to the monkey, which is the promise of a peanut, maybe. And in all these cases, this cell would be active. And they call these the mirror neurons because they are active both for when you perform an action and when you see someone performing the action. So in a way, they were like, okay, this maybe connects, these cells maybe connect our perception of something to when we actually act upon it ourselves. And they were called mirror neurons in a sense that, again, the mirroring of perception to action, I think was the intention to call them the mirror neurons. They had a moment, as I said, even in like philosophical discussions of, you know, brain and whether animals can have a sense of self or something, all those things. I don't think it delivered. So, but if you're interested, you could go look on it. Okay, so one last thing is about what if we have multiple options and we're trying to decide between what actions to take, right? If you have an animal running away from a predator and they see a branching in the road and they try to run away from one, like in one direction, how would they decide that? And in a way, this is a way to close down this sorts of cycle. Like, I think I started the course talking about the perceptual cycle, and the perceptual cycle started from sensation, from the environment, and then perception, attention, and then you make an action, and then you go back into the environment. So this is a schematic of how that could play out, where you have multiple actions that you could decide from, right? The world is always almost giving you multiple choices. Again, another good metaphor for today. But the world gives you multiple choices, and then you pick one of them in this process of basically you have to evaluate what kind of value each of them have to you, right? If you're deciding between chips and something else, and you know, between a salad and a burger or something, and you're kind of analyzing both of them, and then you decide which one to go with. How does that? I think that's the idea. And there was a simple, nice study looking at this. What they looked at, I'm just going to briefly explain this study, is that they were like, okay, if initially you show this to the participant, where you see two spatial cues, one cue on the upper right and one cue on the lower left, and there are different colors. And then you just show the cross, which is the memory period. And then you show the color of the cue that they should attend to. And then this delay comes up, which is the go signal. And then they have to go toward that initial spatial cue that matches the color cue. Well, in this case, because the spatial cue that matches the color cue is red, it's in the upper right, it means that the animal should go to the upper right. Okay, so it's simulating having multiple options. In this case, the options are upper right or lower left. And what you observe is that in this memory period, the animal is keeping both of these options alive. Now, this is in the parietal area, as far as I remember. Do I not have this here? So imagine this is in the parietal area, but I can confirm that later, where you have these different possible attention signals. And what you observe is that a lot of time when you have multiple options in order to act on them, they're both kind of represented at the same time. And then when you make a decision, then one of them goes dead, and then the other one gets much higher activation. So this is, again, it's kind of a generalization of the saliency stuff that I was talking about, that these two options that are salient in the environment. You have to maintain both of them, and then one of them is eventually your chosen thing, and then you go toward that. I think pretty much that's the story here. So the idea here is that as we have these choices to make, when we have multiple choices in the world, and as I said, if you're running away from something, or you're playing a game and you're trying to decide who to pass the ball to, you have multiple options in the world. They're kind of all represented at the same time. And then over this cycle, as you go from the back of the brain to the front of the brain, you evaluate them, and then you kind of rule out some of the options, and then you eventually arrive at one of them, and then you act upon that. That's the idea. All right, that's the last slide. Any questions?